Is CHO still the host of choice? Asked that question 10 years ago, the answer was obvious. CHO is the workhorse of the biopharmaceutical industry. 5 to 10 grams per liter in fed batch routinely. Regulatory agencies have seen hundreds of CHO-derived monoclonal antibodies, and you find CDMO capacity on every continent. The answer was, of course, it is. But ask that question today, and something has shifted. We now see alternative host platforms with real clinical and regulatory data behind them. Not papers, not proof of concept, actual data. Approved products. We have new ones coming in clinical trials. We have regulatory meetings with FDA, MEA, and PMDA. Moss, for instance, has phase 1b data for an enzyme replacement therapy, and the MEA waived human viral testing requirements, which is a regulatory advantage CHO will never have. Plant farming had a fully approved COVID-19 vaccine from Health Canada in 2022. Insect cells are already behind Flublok, Cervarix, and Nuvaxovid. Cell-free protein synthesis has ADC candidates in clinical trials, and it's the only system that can site-specifically incorporate non-natural amino acids, which is exactly what next-generation ADCs require. So, is CHO still the host of choice today? The answer is, it depends on what you're making. And that is a far more interesting answer than the one we had 10 years ago. Welcome to the Smart Biotech Scientist. I'm your host, David Brühlmann. I want to start with a question I've been sitting with for a while. Over the past 2.5 years, we have hosted some extraordinary scientists on this show. People building biologic manufacturing platforms from scratch, using organisms nobody thought belonged anywhere near a GMP facility. We talked about moss, microalgae, silkworms, plants, and cyanobacteria. Every conversation was compelling. Every guest made a strong case. And every time I walked away impressed by the science, but also carrying a nagging question. When you put all side by side and cross-reference them against the published evidence, what does the real picture look like? That's what this episode is about. No guest today, no platform advocacy, just the data, the patterns, and my assessment of where each platform actually stands and where it doesn't. In part 1, I'll walk you through what each platform claims to offer and where the evidence supports or complicates those claims. In part 2, we tackle the big question head-on. Will any of these replace CHO? Let's get into it. Before we talk alternatives, a rigorous look at the baseline is worth it. Chinese hamster ovary cells, or CHO, are the gold standard for glycosylated monoclonal antibodies, and they earned that position. Fed batch titers routinely reach 5 to 10 grams per liter. The glycosylation machinery is well characterized and predictable. Regulatory agencies have cleared hundreds of CHO-derived maps, which means the risk profile is known. The review process is familiar, and the questions are well understood on both sides. And every major CDMO on the planet has CHO capacity, giving you vendor optionality at every stage from early development to commercial production. That combination, high productivity, regulatory familiarity, abundant infrastructure, is hard to displace. And I don't think it's going to be displaced for the products where it already excels. So the relevant question is not whether CHO loses. For standard glycosylated maps with conventional FC function, CHO doesn't lose. The relevant question is, where does CHO leave a gap? There are 3 places to look. Number 1, the cost structure. If a platform can produce the same molecule at a significantly lower capital expenditure or cost of goods, that's the starting point. Number 2, it's speed. If it can take you from gene sequence to first protein in weeks rather than months. And finally, number 3, intrinsic product quality. If a platform naturally delivers a glycan profile or post-translational modification that CHO would require extensive engineering to match. Every platform wins somewhere. The goal of the next few minutes is to figure out where. Let me take you through each platform, what the guests claimed, and what I think is the empirical signal from the published data. The first platform we're going to look at is moss. We covered this extensively in episodes 163 and 164 with Andreas Schaff from Eleva. The claim Andreas Schaff made, and that Eleva has built their entire company around, is that moss offers human-like glycosylation without any animal-derived components. No BSE or TSE risk. That's not a small thing. It simplifies regulatory submissions and eliminates a whole category of safety concerns from your development dossier. But here is the regulatory outcome that I keep coming back to. The European Medicines Agency waived human viral testing requirements for moss-derived biologics. I want you to think about that, what that actually means in practice. Viral clearance studies are expensive. They require often require external testing labs, validated assays, and significant timelines. Skipping that step is not a routine accommodation. Instead, it's a meaningful competitive advantage on both cost and timeline for human injectable products. The second point, moss produces afucosylated glycans natively. You don't engineer it. That's just how the organism processes proteins. For oncology antibodies, where the therapeutic mechanism depends on ADCC, or in other words, the antibody-dependent cellular cytotoxicity, afucosylation directly enhances potency. CHO can be engineered to produce afucosylated MAPs, but that means knocking out the fucosyltransferase gene, validating clone stability through extended culture, and accepting added regulatory complexity. Moss gives you that profile from the start. This is my honest takeaway. For a standard FC intact map where fucosylation level doesn't drive efficacy, CHO remains the better choice. But for an oncology antibody where ADCC is the mechanism, this platform can be your platform of choice. And additionally, Eleva is doing very well in the case of quote-unquote an unproducible protein. If you have a protein that CHO cannot express, moss also can be a very interesting alternative. The second platform is microalgae. We covered this with Muriel Bardor from Alga Biologics in episodes 141 and 142. The sustainability story here is compelling. Photosynthetic biomanufacturing using CO2 as a carbon source is carbon negative by definition. Muriel Bardor's team has demonstrated roughly 70% cost reduction per unit reactor volume, and the oligomannose glycan profile these cells produce is actually favorable for ADCC through enhanced NK cell binding. Now, here is the part I have to be direct about. The yield gap between microalgae and CHO is approximately 1,000-fold. CHO produces grams per liter, microalgae produce milligrams per liter. And I want to say that again, it's not a rounding error. It's the defining technical challenge of this platform, and it has not been solved yet. At the moment, there is no microalgae produced injectable in clinical trials. Alga Biologics submitted a pre-IND dossier to the French ANSM and projected first in human trials for 2026. That is progress and real trackable progress. But a 1,000-fold yield gap means you need roughly 1,000 times the reactor volume to produce the same amount of product. That largely erases the cost advantage, at least at current productivity levels. On the other hand, your microalgae platform is much simpler. It doesn't use expensive media as a CHO culture would do. Here is my takeaway. Watch this platform over a 5 to 10 year horizon because the potential is huge. The sustainability thesis is credible, but we need to solve the yield problem. Platform number 3 is molecular farming. Varnu Pulcharan from Phytofarm presented this technology in episodes 235 and 236. This is the platform I keep coming back to when people ask about pandemic preparedness, and for good reason. The core capability is transient expression in a tobacco-related plant. The number that matters here is this. It takes 3 to 4 weeks from the gene sequence to the first protein. That timeline is not matched by any other platform for complex biologics, not even close. The economic data backs up competitive cost of goods, roughly between $100 to $150 per gram for virus-like particle-based vaccines, which is competitive with CHO for this product class. And then there is COVID-19. Medicago's plant-derived COVID-19 vaccine received full regulatory approval from Health Canada in 2022. Full approval. Phase 3 was positive, plant manufactured, but what happened next is what people cite to dismiss the platform. The product was discontinued because the company's parent company had tobacco industry ownership ties that triggered political opposition in the key markets. The vaccine worked. The regulatory process worked. The manufacturing worked, but what failed was a corporate ownership structure that created non-technical political risk. But dismissing the platform based on what happened at Medicago is like dismissing CHO-based manufacturing because a CHO company had bad quality event. The platform's technical track record is intact, and the manufacturing scale is real. Bahia Phytofarm operates a GMP facility in Bangkok, Thailand, with capacity to produce up to 5 million COVID-19 vaccine doses per month, which demonstrate that plant-based biomanufacturing is not a lab-scale curiosity, but an operational production system. Here's my honest takeaway. For virus-like particle vaccines in pandemic response, plant farming is the fastest path from sequence to dose, with regulatory precedent behind it. The fourth platform is a surprising one, silkworms. Yes, silkworms. I talked about this platform with Masafumi Osawa from CHO in episodes 217 and 218. This is the platform that surprised me the most the first time I really looked at it. One silkworm pupa is functionally equivalent to 100 to 1,000 milliliters of conventional insect cell culture volume. The upstream infrastructure cost is a fraction of a stainless steel bioreactor. Because there's no bioreactor at all. The production model is a living organism. CHO has PMDA alignment for a phase 1 clinical trial of a norovirus virus-like particle vaccine produced in silkworms. They also have a veterinary VLP vaccine that is already registered and commercially used in Vietnam. That is real traction. Here is the challenge though. The batch-to-batch consistency in the living multicellular organism is a QA problem with no established regulatory precedent for human injectables. Every other system we've discussed, CHO, moss, insect cells, and plants operate through a clonal cell populations or at least a tightly controlled plant growth conditions. Silkworm pupae are individual organisms with organism-to-organism biological variability. Managing that variability to meet the batch release specifications that are required for a human injectable is scientifically unsolved territory at commercial scale. My honest takeaway is that the clearest near-term path is both veterinary vaccines and oral delivery formats where the variability tolerances are wider. The PMDA phase 1 alignment is the signal to watch. This is meaningful regulatory progress. The step to human injectables is real and not complete, but a technology to watch out for. Platform number 5, Cyanobacteria. I talked about this exciting organism with Tim Corcoran from Deep Blue Biotech in episodes 229 and 230. Cyanobacteria bring many benefits to the table. We have carbon-negative photosynthetic manufacturing, secretion-capable strains, and potentially very low cost of goods as a long-term proposition. I think these claims are credible as a vision, and there is real progress worth noting. Tim Corcoran's work at Deep Blue Biotech makes a compelling case for where the platform is heading. The ocean-derived cyanobacterial strain they work with, discovered around 2020 off Singapore, is more robust and faster growing than most previously characterized strains. The molecular biology toolkit has matured considerably because of CRISPR-based editing, improved genome annotation, and transformation methods now make the R&D process feasible within reasonable timelines. The direct secretion of the target molecule into the culture medium, so in other words, no cell lysis is required, this reduces cost of goods by an estimated 25 to 35% compared to conventional intracellular production. With respect to the scale up, we see another advantage because photobioreactors, instead of scaling up, they scale out because they are modular glass tube arrays. So a 10,000 liter installation behaves similarly to a 100 liter lab system, which is a meaningful engineering advantage. And the yields are tracking ahead of early projections. Tim described multiples of what they initially thought achievable, which changes the economic calculus for commodity scale applications like biofuels. The near-term commercial focus of their company is high-value specialty ingredients, such as hyaluronic acid for personal care at roughly $2,000 per kilogram, precisely to fund the platform's development toward lower margin, higher volume products. So the current reality is this. We have currently no cyanobacteria-produced injectable drugs that have entered human clinical trials. There are cyanobacterial strains that produce genuine toxins, so-called cyanotoxins, and managing that contamination risk requires rigorous strain characterization and clearance validation that doesn't yet exist as an established regulatory framework for injectables. The strongest near-term applications are biomaterials and potentially oral delivery drugs. My honest takeaway is that this is the most distant from clinical reality of the 5, but it makes a long-term watch and not a near-term manufacturing decision. So let's step back and look at all the 5 together, and if we do that, one pattern becomes very clear. None of them is a universal CHO replacement, not one. Each wins in a specific product category or specific manufacturing context. And the platforms that have made the most regulatory progress, such as moss and plant farming and insect cells, all found their niche first, then built regulatory precedent around it. They didn't try to compete with CHO or on CHO's own terms. Moss didn't try to match CHO titers for standard maps. Plant farming didn't try to compete at commercial antibody scale. They found where CHO leaves a gap, and they built in that gap. Which brings me to the question I want you to carry into part 2. Will it replace CHO even the right question to be asking? I don't think it is. And in part 2, I'll give you the question I think is worth asking instead, and a framework for actually answering it. Thank you for tuning in today, and I'll see you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.